Welcome to DermWire News, powered by PracticalDermatology.com. I'm Stephanie Talea with Practical Dermatology Magazine. Maine is joining 19 other states in allowing students to possess and use sunscreen in school. Governor Janet Mills signed LD 441, which was sponsored by Senator Kathy Breen. Modeled after American Society for Dermatologic Surgery Association's Sunucate legislation, the bill was designed to eliminate barriers that prohibit students from possessing and using over-the-counter sunscreen in school by exempting these products from broad-reaching medication bans. Students in Maine will no longer need a prescription or physician's note in order to apply sunscreen during outdoor activities. The need for this legislation stemmed from dermatologists and reports in the national media that children could not use sunscreen while outside unless they have a doctor's note or prescription. Although sunscreen is classified as an over-the-counter medicine by the FDA, the Centers for Disease Control and Prevention and the United States Preventative Services Task Force both believe that children should have access to sunscreen and other sun protective measures in order to reduce the risk of skin cancer. ASDSA worked with the Maine Dermatological Society and the Personal Care Products Council to advocate for the passage of this law. To learn more about Sunucate, visit asds.net/SUNUCATE. InMode Limited is introducing Accutite, the smallest radiofrequency-assisted lipolysis or RFAL device for precision fat reduction or skin contraction in the face and body. With its sub-millimeter cannula, Accutite enables focal RF contraction without excisional surgery. There is little or no visible scarring and minimum downtime with Accutite. The procedure takes less than 15 minutes under local anesthesia. Results can be seen immediately with continuing improvements for several weeks following treatment. These results are maintained for up to 12 months post-RFAL procedure. Accutite now joins InMode's RFAL technologies. Moffitt Cancer Center researchers have discovered a mechanism by which melanoma cells become resistant to drugs that target the BRAF protein and its signaling pathway. Their study was published online in the journal Cancer Research. Mutations in BRAF are found in approximately 50% of all melanomas, leading to increased cell proliferation and survival. Several drugs are available that target BRAF and a downstream gene named MEK that cooperates with BRAF in cancer development. These drugs have resulted in significant improvements in patient outcomes. However, many patients eventually develop drug resistance. In a series of laboratory experiments, with cell lines and mouse models, researchers discovered that melanoma cells that are resistant to BRAF inhibitors undergo a similar response mechanism as cells treated with stress-inducing factors such as UV radiation and low oxygen. During each of these conditions, the cells increased expression of the protein histone decetylase 8, HDAC8. HDACs are proteins that regulate the expression level and activity of other proteins within the cell. HDACs are often deregulated in cancer, and several drugs that target HDACs are approved for the treatment of different types of cancer. The researchers found that HDAC8 stimulated the activity of the AP1 transcription factor that subsequently increased the expression of genes that are involved in cell motility and invasion. Our work provides the first evidence that HDAC8 activity is increased in response to multiple diverse cellular stresses, and that this in turn initiates a transcriptional program that is associated with increased melanoma cell survival, explains Dr. Kieran Smalley, director of the Donald A. Adams Melanoma and Skin Cancer Center of Excellence at Moffitt, in a news release. The link between HDAC8 and drug resistance suggested to the researchers that drugs targeting HDACs may be able to overcome cellular resistant mechanisms to BRAF inhibitors. The researchers performed a series of mouse experiments and found that co-treatment with both HDAC and BRAF inhibitors inhibited melanoma tumor growth better than either agent alone. The researchers hope their work will lead to clinical studies that will examine the potential of HDAC inhibitors to prevent the development of drug resistance in melanoma. These findings provide a strong rationale to pursue the development of more selective and potent HDAC8 inhibitors for future evaluation as drugs that can limit phenotype switching and therapeutic escape in melanoma, says Dr. Smalley. Thank you for listening to DermWire News, powered by PracticalDermatology.com.